Hi students, coming to the next topic is DNA replication process. Before going to discuss about DNA replication process, we already discussed autocatalysis process. DNA replication, that is called as autocatalysis process. See, synthesis of DNA from DNA. Synthesis of DNA from DNA, that is called as DNA replication process, that is also called autocatalysis process. So today we are discussing about DNA replication process. Before going to discuss about the DNA replication process, okay, we have to discuss about enzymes. Means the enzymes which are involved in DNA replication process. The enzymes which are involved in the DNA replication process. So this is also called enzymology of DNA replication process. This is also called enzymology of DNA replication process. So a group of enzymes are there, the enzymes which are involved in DNA replication process. So this is called as enzymology of DNA replication process. And all we know the cell cycle. All we know the cell cycle. Within that cell cycle there is a G1 phase is there, S phase is there, G2 phase is there. G1 phase is there, S phase is there, and G2 phase is there, followed by karyokinesis and cytokinesis. Karyokinesis followed by cytokinesis. Today here we are discussing about there is a G1 phase is there, G1 phase and S phase is there. Exactly in S phase DNA replication is takes place. In S phase DNA replication is takes place. But here whatever the enzymes are there, the enzymes which are required for the DNA replication process, the enzymes which are required for the DNA replication process, all these are synthesized in G1 phase. All these are synthesized in G1 phase. The enzymes which are required for the DNA replication process, all these enzymes are, majority of the enzymes are synthesized in G1 phase, right? So that is about that synthesis of enzymes which are required for the DNA replication process. Most of these enzymes are synthesized in G1 phase. In S phase, DNA replication takes place. So today, first of all, in this class, we are discussing about enzymology of DNA replication. Means a group of enzymes which are involved in DNA replication. A group of enzymes which are involved in DNA replication. So in that case, the very first one is the topoisomerases. Topoisomerases are the enzymes. Topoisomerases are the enzymes. See again here two types of the topoisomerase enzymes are there. There is a two types of topoisomerase enzymes are there. Based on the function, these are classified. Based on the function, these are classified. The very first one is topoisomerase 1 and next one is topoisomerase 2. The very first one is topoisomerase 1 and next one is topoisomerase 2. Here topoisomerase 1, that one is indicates, see the enzyme especially involved in positive supercoiling. Positive supercoiling. See, positive supercoiling means whatever the DNA is there, DNA forms the coils like structures or helix like structures. The DNA forms the coils like structures or helix like structures. For suppose this is the DNA. This is the DNA, right? This is the DNA. Generally during condensation process, helix like structures are formed. The DNA forms the helix like structures. So here these are the helix like structures. So this is the double stranded DNA. This is the double stranded DNA. During DNA replication process, if these helix are present, the DNA polymerase enzymes are unable to move on DNA templates. If the helix like structures are there, presence of these helix, the enzymes which are involved in the DNA synthesis, which are unable to move on DNA templates. So a group of enzymes which are involved in the removal of helix. So this is the double stranded DNA. The helix are removed. So removal of helix done by topoisomerase 1. Whatever that DNA contains, tension is there or what? This is the tension state. Right? So the tension removing or the tension relaxation done by topoisomerase 1. The tension relaxation is nothing but helix like structures. Whatever that helix like structures are there, the helix like structures are removed by the topoisomerase 1. So this is called as positive supercoiling. Positive supercoiling indicates the DNA contains helix-like structures are removed. Helix-like structures are removed, or tension, relaxation done by the topoisomerase 1, right? So this is nothing but unwinding DNA. Unwinding DNA means here not the separation of two strands. Here whatever that coils-like structures are present, the coils-like structures are removed by the coils-like structures are removed by the topoisomerase 1, right? And coming to the next one, supporting for the transcription. See the very first one involved in DNA replication process, right? See, coming to the next function is transcription process. During transcription also, see, transcription means what? Synthesis of RNA from DNA. Synthesis of RNA from DNA. See, during the transcription process, whatever the DNA, again here also same function, coils-like structures are there, the coils are removed. Once the coils are removed, and the DNA involved in open bubble formation. Open bubble formation. Once the open bubble formation takes place, then RNA synthesis will be takes place. So here the transcription supported by the topoisomerase 1. And coming to the next one is the recombination process. We know that recombination is nothing but exchange of genetic material, especially that is takes place in the meiosis process. Within that meiosis, there is a packeting stage is there. Within that packeting, between that two chromosomes are there, especially two non-sister chromosomes are there, right? Whatever that homologous chromosomes are there, between that homologous chromosomes, exchange of genetic material takes place. So what I am telling here, the chromosomes contains DNA, shifting from one chromosome to another chromosome. See, the homologous sequences are shifting from one chromosome to another chromosome. The process is called as recombination. So the recombination also supported by the topoisomerase 1, because here relaxation of DNA. So in all the cases, either DNA replication, either the transcription, either the recombination, all these cases, the topoisomerase 1 involving relaxation of DNA or removes the helix-like structures, right? Coming to the next one is that topoisomerase 2. Within that topoisomerase 2, here there is a negative supercoiling. This is positive supercoiling, this is negative supercoiling. See, the difference is here, one is that relaxation of DNA. I said here this is the coiled DNA, relaxation. So the relaxation done by topoisomerase 1. And other one is generation of tension or formation of coils-like structures. So the topoisomerase 2 involved in negative supercoiling process. So that one involved in the winding process. Winding is nothing but, this is the DNA after DNA replication. This is the state of the DNA, right? Again here helix-like structures formation. So one is removes the helix-like structures. Here second one is involved in the helix-like structures formation. So here one is that unwinding DNA, other one is the winding. Then what is the use of topoisomerase 2? We know that chromatin, right? Free state or relaxed state of the DNA. See how the relaxed state DNA forms the chromosome by the condensation. During condensation process, whatever that topoisomerase 2 enzymes are there, these enzymes are supporting for the coils-like structures formation. That coils-like structures are helix-like structures supporting for that nucleosome formation. That nucleosome supporting for that again solenoid formation, supersolenoid formation, scaffold formation, right? Chromatin formation. So here all these are the functions of the topoisomerase 2. So in this concept, what I am telling regarding to that topoisomerase 1 is supporting for the relaxation or removes the tension from DNA. Means removes the coils-like structures. So supporting for DNA replication, supporting for transcription, supporting for the recombination. And next one is, other one is here negative supercoiling is there, right? Here whatever that coils-like structures, after that DNA replication process, here coils-like structures are generated or helix-like structures are generated. winding process, and chromatin condensation process, means chromosome formation, right? That is about that topoisomerase 1, topoisomerase 2, functions of topoisomerases, right? Coming to the next topic is the DNA helicase enzyme. See, in the first case, means the very first enzyme we discussed about the topoisomerase, right? And coming to the next enzyme is the DNA helicase. So what I am telling here, DNA helicase enzyme, that enzyme supporting for the separation of the two strands of the DNA, means okay, this is the one of that DNA. Okay, this is one of the DNA during DNA replication process. First of all, the coils-like structures are removed. Okay, right? We already discussed about the topoisomerase enzyme. And next one, the closed strands are open. The closed strands are open. See, whatever the DNA is there, between the two strands of the DNA, there is a hydrogen bonds are present. That hydrogen bonds are cleaved by one of the enzyme. The enzyme name is DNA helicase. DNA helicase is the enzyme. That enzyme involving the cleavage of hydrogen bonds. Cleavage of hydrogen bonds. Once the hydrogen bonds are cleaved, there is an open bubble formation is take place. So all these are indicates hydrogen bonds. The hydrogen bonds are cleaved by the DNA helicase enzyme. Once the hydrogen bonds are cleaved, there is an open bubble formation. There is an open bubble formation. Simple. Whatever that double helical DNA is there, this is the double helical DNA, right? That this is the double helical DNA. That double helical DNA contains the hydrogen bonds are there, and these hydrogen bonds are cleaved by the DNA helicase enzyme. Then there is an open bubble formation is takes place. There is an open bubble formation is takes place. Once the open bubble formation is takes place, the DNA polymerase enzymes are binded and involved in the DNA synthesis process. So we need to know about the characteristic features of the DNA helicase enzyme. This is one of the motor proteins. A group of proteins are there which are moving on DNA, which are moving on the DNA templates, So motor proteins, hexameric proteins, means totally 6 proteins. 6 protein molecules are combined. So that's the reason why hexameric proteins means totally 6 proteins are combined and involved in the hydrogen bonds cleavage. That group of enzymes collectively called as DNA helicase enzyme. That hexameric protein structure binds to the DNA template, right? This is the one template, this is another template. Hexameric proteins are binded to the one template, right? And hydrogen bonds are cleaved. This is the DNA template. Hexameric protein is binded. See, this one is moving on DNA. This one is moving on DNA. Whatever that hydrogen bonds are present between the two strands. The hydrogen bonds are cleaved. Once the hydrogen bonds are cleaved, the two strands are separated. Right? So this enzyme requires energy in the form of ATP for the cleavage of hydrogen bonds or for the movement of DNA helicase enzyme. Requires ATP. So presence of utilization of ATP, the enzyme moving on the DNA templates cleaves the hydrogen bonds and the two strands are open. The last point is cleaves the hydrogen bonds. We studied. Okay, see, we know about this one because whatever the two strands are there, between the two strands, the hydrogen bonds are cleaved. That is about the DNA helicase enzyme. Right? Coming to the next topic is that SSB proteins. So before this one, we discussed about the topoisomerase enzymes and the DNA helicase enzymes. And coming to this topic, single-stranded binding proteins. SSB indicates here single-stranded binding proteins. A group of proteins which are supporting to prevent the reassociation of the DNA. How the process takes place, we will discuss now, right? Here, this is one of the double helical DNA. See, the DNA contains that helix-like structures. The helix-like structures removed by the topoisomerase enzyme. And coming to the next one, DNA helicase enzyme. That enzyme binds to that DNA template and involving the cleavage of hydrogen bonds. Once the hydrogen bonds are cleaved, the two strands are separated, and these two strands are separated. After the separation, the reassociation of the two strands prevented by SSB proteins. So why the DNA have the reassociation capacity? Because the DNA having always complementary base pairing. One strand contains adenine, other strand contains thymine. One strand contains the guanine, other strand contains the cytosine. So what I am telling here, whatever the two strands of the DNA are there, the two strands are having complementary base pairing. Due to that complementary base pairing, right, DNA naturally have the reassociation capacity. That reassociation prevented by the SSB proteins. A group of proteins are binds to the two strands of the DNA and prevents the hydrogen bonds formation. So here, after the separation of two strands, prevents the recoiling or reassociation of the DNA, right? And DNA have the negative charge. We know DNA have the negative charge, and histone proteins have the positive charge, right? That's the reason why there is a condensation takes place. Here also same thing, DNA have that negative charge, and SSB proteins, single stranded binding proteins, have the positive charge. That's the reason why between these two there is an association takes place. And prevents the hydrogen bonds formation. The simple explanation. See, this is one of the thread-like structure. There is a white color thread-like structure is there. That is the DNA, and this is the separated. Two strands of the DNA. These are the strands, separated DNA strands. The beads-like structures. Here the beads-like structures indicates SSB proteins. The beads, each blue color bead is there, right? Each bead indicates here SSB proteins. So the SSB proteins prevents the hydrogen bonds formation. Once the hydrogen bonds formation is prevented and there is no chance for the reassociation of DNA, then what is the prevention of the reassociation? Once that open bubble formation takes place, then the DNA replication takes place. If the two strands are closed, there is no chance for the DNA replication. So if the two strands are open, there is a chance for the DNA replication, right? That is about the SSB proteins, right? Coming to the next enzyme is the RNA primase enzyme. RNA primase enzyme involved in synthesis of RNA primers. Then what is the use of RNA primer in DNA replication process? See, we know within that deoxyribose sugar or ribose sugar, the third position is very important. This is the first position, interact with nitrogen base, right? This is the second position. This is the deoxyribose sugar, hydrogen atom. Here also hydrogen atom. This is the third position, hydroxyl group is present. This is the third position, hydroxyl group is present. So what I am telling here, that hydroxyl group is very important. This hydroxyl group is very important for the polymerization process or DNA replication process. So for that purpose, this is the one strand and this is one strand. After that, the coils removing process. After that, hydrogen bonds cleavage. After the binding of single stranded proteins. Right. The next action is RNA primase enzyme. Here one of that. This is one of the RNA primer. RNA primers are synthesized. What is the use of RNA primer here? It provides the hydroxyl group. Once the hydroxyl group is available, the new strand is synthesized. One of the strand is continuously synthesized. The strand is called as leading strand. Leading strand always requires one primer. Right? Coming to the next one. Lagging strand is there. There is a bits like DNAs are synthesized. So means the lagging strand synthesizes the DNA in the form of bits like structures. These are called as Okazaki fragments. So within lagging strand, the DNA is synthesized in bits manner. So every bit requires one primer. Every bit requires one primer, or every Okazaki fragment requires one primer. So here every Okazaki fragment, okay, requires one primer, and each primer provides the hydroxyl group. Each primer provides the hydroxyl group. So what I'm telling here, one of the strand is continuously sensile. The strand is called as leading strand. The leading strand requires only single primer. Single primer. This is the primer. Okay, composition of the primer is the RNA. Composition of the primer is the RNA, right? And this is the another strand. The strand is called as lagging strand. Lagging strand sensizes many number of bits-like structures. The DNA sensized many number of bits-like structures. Each bit is called as Okazaki fragment, and each bit requires one RNA primer. Each bit requires one RNA primer. So for the synthesis of lagging strand, many number of RNA primers are required. Many number of RNA primers are required. So the function of RNA primase enzyme synthesizes the RNA primers. That RNA primers provides the 3' hydroxyl group. 3' hydroxyl group involved in the polymerization of DNA. Polymerization of DNA, right? Remember, this is the DNA bit. The black color indicates here DNA bit. The red color indicates here RNA. Once the DNA replication is completed, replication is completed, again the RNA fragments are removed. The RNA fragments are removed. Again here new DNA bit is sensized. New DNA bit is sensized. After DNA replication process, RNA primers are removed. After DNA replication process, okay, the RNA primers are removed. Again DNA synthesis takes place. Okay, that is about the RNA primase enzyme. And listen all of you, involving RNA primer synthesis. RNA primase enzyme involving synthesis of RNA primers. RNA primers provides the 3' hydroxyl group. 3' hydroxyl group involving polymerization process. After DNA replication process, RNA primers are removed. Right? And in that place again DNA synthesis will take place. That is about the RNA primase enzyme, right?